Hi, everyone. All right, so up to this point in biochemistry, we have now categorized a bunch of different kinds of molecules. We've got inorganic, organic, we've got carbs, lipids, proteins. Now we're going to turn our attention to one special, really important category of proteins called enzymes. You are alive because of your enzymes. You would cease to exist without your enzymes. I want you to think back to what I said at the beginning of this chapter. You are a walking, talking pile of chemical reactions. Your dehydration, synthesis, and hydrolysis and respiration. You're doing all kinds of chemical reactions where you're moving atoms back and forth. And that's really what you are. You are all these atoms interacting with each other in really cool ways, and they make up your cells that make up your body. Well, chemical reactions don't just happen. You don't just magically get cell respiration to happen. If it did, rocks would be doing cool things or something like this. You just have magically living stuff. And that's not what happens. Life is special, and it doesn't just happen among any old atoms. The reactions that happen in your body are very specific and only happen because you have enzymes that make them happen. And so really the core substance of being alive is the chemical reactions, and they are controlled by enzymes. So these are called catalysts. They are protein catalysts. A catalyst is anything that controls a reaction without being changed itself. And in this case, it's a catalyst made of protein. And the way it works is you have an enzyme that you've made in your cells, and you've got hundreds of thousands of enzymes all in your cells, and you have one that you've made, and it will attach to whatever the reactants are in the reaction and force them together or tear them apart. But this enzyme, this protein, will actually, by its shape, physically attach to something and then force it together or tear it apart. So if you look, if the blue part here represents an enzyme and the yellow part represents the thing that's going to be a part of the reaction, we'll call it a substrate. That is the thing it acts on. If the blue is the enzyme and the orange or yellow is a substrate, what happens is it fits right in like a puzzle piece. If the enzyme's the wrong shape, it won't fit. So enzymes are very particular to the substrates they act on. And in this case, the reaction would be to start with this two pieces hooked together, and the enzyme forces them apart, and we have the products of the two pieces. So again, the substrate fits exactly to the enzyme, and it forces the reaction, in this case, to break them apart. This reaction might be taking a disaccharide, right? We've got two rings here hooked together, and this enzyme is forcing it down into two monosaccharides, and that would be hydrolysis. So this might be an enzyme that causes hydrolysis of this particular disaccharide, okay? And there are many, many different enzymes, and they each control different reactions. So if you look at this one, perhaps this is an amino acid, and this is an amino acid. Here's an enzyme, a protein in a particular shape that will allow this to fit in here and this to fit in here. And these two pieces of the substrate will be brought together and leave as one. So we might have two amino acids being put together into a dipeptide by dehydration synthesis. And so this enzyme's job would be dehydration synthesis of those two amino acids into a dipeptide, okay? And the shape of each determines its function. So if an enzyme is shaped like this, it's not going to work on this substrate. It's only going to work on this substrate. And so you have specific enzymes that you make for particular reactions. If you want to do hydrolysis of a disaccharide, you're going to produce this enzyme so that you can do that reaction over and over and over in all your cells. If, on the other hand, you want to put amino acids together into a dipeptide, you're going to produce this enzyme so that you can do that over and over and over. And that's how you're a living being. You control what you're doing by controlling which enzymes you make. So I'm going to back up for a second. So one of the key things here is if you look at the enzyme at the beginning and the end, it's exactly the same. It's not part of the reaction. No atoms are being taken off of it. No atoms are being added to it. Same thing up here. It's in the same exact shape as when it started. Enzymes are not changed by the reaction. Only the substrate is. Okay, the substrate, in this case, at this point, the reactants, and here the products, that's changed. That's shifting atoms around. That's splitting apart or coming together or doing something. The reaction is with the substrate. The enzyme is not part of the substrate. It is not changed by the reaction. Okay, and the shape of the enzyme is critical. If the enzyme's shape changes for some reason, it won't work. So it's really important that it be a specific shape. Frequently, the names of enzymes end with ASE. Do not confuse that with OSE, which is common end to names of carbohydrates, right? So if it's ASE, you have a little clue that it's probably an enzyme. That is a protein that controls reactions by matching to the substrate and by shape and causing the reaction to happen without being changed itself. And that, I think we'll get into our notes real quick right here. Okay, so enzymes attach to the substrate, causing it to be broken or combined, and the reaction occurs, and the enzyme is unchanged. Enzymes work because their shape fits exactly to the substrate.
Reactions do not occur spontaneously in the cell. Instead, cells rely on proteins called enzymes to kickstart chemical reactions and speed them up, enabling cells to get the most out of the energy sources available to them. Enzymes have a unique way of kickstarting reactions. They work by binding to one or more specific molecules called reactants, or substrates. Binding occurs at a special region of the enzyme called the active site.
The active site is where the actual substrate fits in. So its shape is the absolute critical part.
Once the substrates bind to the active site, they form what's called an enzyme-substrate complex. As the enzyme and substrates begin to react, some of the chemical bonds in the substrates begin to weaken, causing them to link together. Eventually, the chemical reactions at the active site lead to the formation of a different molecule. This is referred to as the product. Once the reaction has occurred, the product is released from the active site. The enzyme returns to its original state and is free to react again with another set of substrates.
And so the enzyme can be reused and reused and reused. It's like a little factory within your cell, and so critical for the functioning of all of your cells because it controls all chemical reactions.